Here are the video notes for part 2 of chapter 14, section 1. So we're continuing our look at human genes. How are they passed on, and how are the different characteristics ranging from the color of your eyes to specific proteins within your cells, how are those passed down? This section is going to look at specific kind of disorders or genetic information on the autosomal chromosomes. And a reminder, the autosomal, those are the ones not involved in determining the sex of the organism. So the autosomal are those, the other 44 chromosomes that are not related to that. I'm just going to change this here. Hopefully you can see that. One that we're going to look at is blood type. So when we think about if you get injured, you lose a lot of blood, you go into the hospital to get a blood transfusion, but it's not as just easy as going in there and say, hey, give me whatever blood I need, whatever blood you have. Our blood type has a bunch of different genes involved, and if you get the wrong blood type, there can be serious complications. So we're going to look at how is that genetic information, what do we know about it? Luckily for us, blood type was one of the first human genes to actually be identified. And there's two main ones that we're going to look at when we talk about kind of the type of blood type that you have. The first one, I'm going to put a number 1 by it, is the Rh. Often you'll hear like Rh factor. And this is kind of the blood group. This part of the blood group is determined by a single gene with 2 alleles. You guys have seen this. You've done it in chapter 11, our typical Punnett square. Our positive is what we would call our dominant allele, and if you're negative, you contain both 2 recessive alleles. And so in our positive, you're going to see both. You're going to have 2 positives or a positive and a negative, and for the negative, you'll actually see both negative for that. Again, you could give them, you know, a capital R and a little r and kind of go about it that way. But you're positive if you have any dominant, and you're recessive, you're negative if you have both recessive. And this came from actually a name for rhesus monkey, and this is the animal that these alleles were discovered in, and kind of gives us just a little bit more information about your genetic code. So why does this matter? Why does it matter if you have positive? Why does it matter if you have negative? Well, this we see a lot with pregnant women or moms who might have more than one child. So what happens is these are not compatible. If you are given blood and you're positive and you're given negative blood, your body is basically going to create antibodies to break that blood down. So that's why it's important to understand, what do I have? So how does this play a role with pregnant ladies? Well, what will happen is with their first child, if they're different, suppose the mom is Rh negative and they figure out that the child is Rh positive. Again, her body's going to start creating these antibodies, and if she has another kid, these antibodies are going to go and kind of attack that fetus. So it plays a role in pregnancy, and that's why a lot of pregnant mothers will actually have a test to determine what is their Rh factor. So that's one piece of the puzzle. The other piece of the puzzle that we're interested in is the ABO blood group. And this helps us, again, determine that actual blood type. And there are 3 possible alleles in this group. So we know that we're dealing with multiple alleles, and we're going to have this combination of codominance and recessiveness. So if you have IA and IB, these would be considered codominant, I is recessive. And how we often see this is we might see IA, IB, and little I for our 3 possible alleles. And these are determined by the type of antigen that they will produce. Antigens are kind of proteins located on the surface of red blood cells, and depending on the different blood type, they're actually going to produce different antigens. So it's important again to understand what is the blood type so we know what type of antigens are being produced. So the IA is going to produce A antigens, and we'd consider them type A blood. IB is going to produce the B antigens, and we get the type B. And a combination of both of those is going to give us AB. Again, that idea of the co-dominance. Two little i's, do not actually produce antigens, and we will call this type O blood. So, if you hear AB negative, what this really means is you are going to have the co-dominance, so you're going to have one, you're going to be heterozygous with one of each, IA and IB, and then you're going to have 2 Rh negative alleles from the Rh gene. So this kind of gives you an idea of when we're saying this stuff, what do we actually mean? And this little chart here kind of shows you a little bit more about what we would call the blood type when we're just dealing with the A, B, and O. This chart unfortunately doesn't have the negatives and positives involved with it. But as you can kind of see, where can people receive their blood? Notice for O is involved with all of these. So anyone who has A, B, or O can all receive blood from O with minimal complications. And that's why we look at the blood type for that. Just a little screenshot here. What are the most common blood types in the US? Again, notice our O positive blood type in the middle here. And the nice thing about that, again, O is kind of the universal donor for all the other blood types. It kind of makes sense that we're positive versus negative, that dominant allele for positive. In a close second, we have A positive, and then B positive is quite a bit behind in there. But if you notice, a lot of our negatives have a little bit smaller percentages for that. But this just gives you an idea of where, what blood types are kind of common in the US. Okay, the last kind of little piece that we'll do with this, again, just this idea of predicting the outcomes of genetic pairings, genetic crossings. So if you have a woman who has heterozygous type A blood, okay, remember, heterozygous, we're going to have 2 different alleles, and it's for type A. So we know that she must have a big I with an A and a little I, because we could not have this because of the co-dominance. This would tell us she was type AB, and we know that that is not the case. So this is why we can determine the genotype just from that information that was given in the question. All right, we also have, a man with type AB blood. Again, because of the co-dominance, we're going to see IA and IB. And we're just going to do our typical Punnett square that you guys have been doing now for a couple of weeks that you did in chapter 11. And this will give you an idea of what are the possible outcomes when these are crossed together. So we're actually looking at the blood types. What type of blood types are we going to get? Now think about what is the difference between the blood types and the genotypes for the blood. blood types are going to do the phenotypes. So type A blood, we are actually going to get 2. I'm going to call it from right here. We get both of those for type A. We have a type AB. We have one of those, which comes from this one right here. And our last one is going to give us type B. If at this point you are still not comfortable with Punnett squares, make sure you reach out and talk to me. This ends part 2 of chapter 14, section 1, giving you just this quick little idea of one of the autosomal genetic traits that we are going to be interested in how they are passed on. In the next portion of the notes, we will go into more of these, starting with some of the recessive alleles and the genetic kind of disorders that you can get by the passing on of these recessive alleles.